Hi guys, welcome to my YouTube channel Unique Bio Classes based on NCERT syllabus for PUC first and second year. If you are new to my channel, please like, subscribe, share and hit the bell icon so that you can get notifications as soon as videos are getting uploaded. Okay, we shall start today's class. Okay, so in previous video we have discussed about pre-fertilization structure. We have discussed about structure of a flower, pre-fertilization structure and events, and we have started with the stamen, microsporangium and a pollen grain. Okay, we have discussed about the stamen. As if we overlook that, stamen consists of two parts. One is anther, another one is filament. Filament is the longest part. Anther, it has two lobes, and each lobe it has diathecus, and inside that, when you take a transverse section of it, you will find it is a four-sided, so it is tetragonal, and each one side it is having a microsporangia. So totally it is having 4 microsporangia, right? We were saying this. Later these microsporangia will produce pollen grains, and these pollen grains are stored in these tubes called as pollen sacs. Earlier we have called it as this one four-sided lobes, but now we are saying it as pollen sacs when it is started to produce pollen grains. Okay, after this we have discussed about structure of microsporangia. When we have discussed about structure of microsporangia, we said it consists of 4 layers. One is epidermis, endothecium, middle layer and a tapetum. The first 3 will provide protection and tapetum will provide nourishment for growing pollen grains. Tapetum is having a dense cytoplasm and it has more than one nucleus. Because it is having many nucleus, so it is used for nourishment. Okay, then after this we have here, we have stopped till here. We shall proceed further. In my further video you will find about this one. We shall see microsporogenesis, right? Hope you are able to find microsporogenesis. This microsporogenesis, one second. Okay, hope now it is visible. Microsporogenesis, it is the process of formation of microspore from microspore mother cell through meiosis. This will be a definition of microsporogenesis. Anyhow, I shall discuss about that one later. What is the definition of it? Here the cells of the sporogenous tissue, as we have discussed in the other class in the previous video, that these were the 4 layers, and this is the microsporangium, which is covered by tapetum, and it consists of sporogenous tissue. When anther is young, it consists of a sporogenous tissue. Now each sporogenous tissue will develop into microspore tetrad. Okay, so that if I show you here, if I am drawing this as microsporangium, one of that microsporangium I am drawing it here, it consists of sporogenous tissue. This sporogenous tissue is nothing but cells. Okay, it consists of number of cells, and each sporogenous tissue is capable of producing microspore tetrad. Now what is this microspore tetrad? I shall make you understand. Now each sporogenous tissue is later, as it is going to develop, sporogenous tissue will get converted into pollen mother cell, or it is also called as gamete mother cell or microspore mother cell. Okay, when anther is young, it consists of sporogenous tissue. As anther is getting matured, this sporogenous tissue will get converted into pollen mother cell or gamete mother cell, which means each sporogenous tissue is capable of producing pollen grain. Now gamete mother cell is nothing but, we said it as, it is also called as meiocyte. In your first chapter we have discussed. Okay, this meiocyte is nothing but diploid. Gametes will be haploid. Now these pollen mother cells will undergo meiosis. Okay, or microspore mother cell will undergo meiosis to produce microspore tetrad. Okay, this microspore tetrad will be like this. From each one sporogenous tissue, it will form a microspore tetrad, which means one sporogenous tissue is capable of producing 4 pollen grains or 4 microspores. Pollen grain is also known as microspore. Now this one is microspore tetrad. Where it will be produced? It will be produced inside this. It will be like this, and you can find this kind of microspore tetrads are present. How it has been formed? Because of meiosis. This diploid will get converted into haploid microspore tetrad, which means each microspore is haploid. Okay, now as anther is getting mature, okay, and it gets dehydrated, these microspores which are present inside that, those will get separate or dissociate with each other. Okay, dissociate with each other, so that what they are in the groups as 4, now they will get separated. Okay, now they will get separate. Now each one we call it as pollen grain, or it is also called as microspore, or it is also called as microspore. Okay, pollen grain or microspore. When they are in groups of 4, we call it as microspore tetrad. When they are alone, when they have been separated, we call it as microspore or pollen grain. These microspore tetrad have been formed from what? These have been formed from gamete mother cell. This gamete mother cell or pollen mother cell is formed from what? From sporogenous tissue. So how many microspore tetrads, how many microspores are produced from one sporogenous tissue? Can you answer this question? How many microspores are produced from one sporogenous tissue? Already I gave the answer. Hope you will try to get the answer properly. Okay, this is how it produces. Now, what is microsporogenesis? You will find it here. The process of formation of microspores from microspore mother cell or pollen mother cell through meiosis is called as microsporogenesis. This will be the definition for microsporogenesis. Okay, hope you understood how pollen grains have been formed. Now this is the mature anther. Okay, so this was young anther, this is mature. Now it has undergone dehiscence. Dehiscence, we said it is nothing but getting a simple cut over it. So the structure which was something like this earlier, now it got a cut over it. So how it appears now? It is appearing something like this. Okay, in here pollen grains which were present in the this one pollen sacs, those will be released out. Okay, this takes place after dehiscence. So this is the mature anther, mature or dehisced anther. This is the young anther, and this is the elaborated structure of it. Okay, once again, if I'll discuss about that, just we will go through an overview of it. Sporogenous tissue, each sporogenous tissue as anther is getting mature will get converted into pollen mother cell or microspore mother cell. These pollen mother cell or a microspore mother cell, they are diploid because they are meiocyte or a gamete mother cell. These pollen mother cell will undergo meiosis to produce microspore tetrad, because as you know, one cell when it completes meiosis, it produces 4 cells. Is it? When one cell it completes meiosis, it produces 4 cells, 4 daughter cells. Whereas in mitosis, when one cell completes mitosis, it produces 2 daughter cells. Okay, so you know this. So as soon as meiosis is completed in pollen mother cell, it produces 4 microspores. That's why it is called as microspore tetrad, and each one is haploid. From one sporogenous tissue, 4 microspores will be produced. Okay, now these microspores which are in group of 4, now they will dissociate with each other when that anther matures and dehydrates. At that time, these microspore tetrads will dissociate and it forms pollen grains. These pollen grains or microspores will be released out, and this after getting released from the dehisced anther, these pollen grains will be undergone pollination and further proceeds with the fertilization. Hope you understood till here. Okay, this is about microsporogenesis. Okay, then we shall discuss about pollen grain. If we go through here, first we talked about stamen. Now microsporangium is completed. Now the last one is left is pollen grain. Okay, in a pollen grain you will find the pollen grains. Here I have mentioned pollen grain is also called as microspore. The pollen grains represents the male gametophyte. Male gametophyte is what? Gametophyte is that which carries gametes. Gametophyte are mentioned for those which carry its gamete. So here you can find it's a male gametophyte, means pollen grain carries male gamete. Pollen grain is not a gamete, it carries a male gamete. Okay, so pollen grain is haploid, it is unicellular body and single nucleus. So these pollen grains, whenever you touch a flower, sometimes you might have noticed that your fingers are attached with some of the powdery substance. Those powdery substance is nothing but pollen grains. Okay, you can put it on the slide with 1 drop of water, and you can observe that one under microscope. So you'll find different types of structures of pollen grains. The size, shape, color of the pollen grain varies, as you can find in your textbook, something like this. All these are different types of pollen grains. Okay, now this diagram which they have shown, it is nothing but microspore tetrad. Okay, microspore tetrad. Later it will get dehydrated and each one will get separated. Okay, now structure of pollen grain. In the structure of pollen grain, they have asked for 2 marks only to draw, for 3 marks for explanation. So you must know every detail of it. Okay, first I shall draw the structure of pollen grain. Okay, I'm just drawing roughly. Okay, this one is exine, intine, and then you have two cells. One is vegetative cell, another one is generative cell, and this one part which I am making it dark, that is called as germ pore. Okay, so this is the structure of pollen grain. Okay, you should draw it neatly, and then it is filled with the cytoplasm. Fine. So this is the structure of pollen grain. It consists of exine, intine, and it consists of 2 cells. One is vegetative cell, another one is generative cell, and there is presence of germ pore. This cell consists of cytoplasm. Okay, here you can find exine. Exine is the outermost layer. This one is the exine. This exine is made up of sporopollenin. Sporopollenin is nothing but an organic material which is resistant to all kinds of chemicals, acids, alkalis, and even you don't have one such kind of enzyme which is going to degrade this. Okay, it is an organic material which is resistant to all kinds of acids, alkalis, and no such enzyme is available for degrading this. Because of presence of sporopollenin, this pollen grains can be stored for a longer time without making it get damaged. It will be stored for a longer time without getting a single damage for this pollen grain, because of presence of this kind of chemical called as sporopollenin. Sporopollenin is used in the formation of exine. It is very hard. It is it is resistant to different kinds. If you take the pollen grain and put in one kind of any one of the acid, it does not happen anything for the pollen grain because of presence of sporopollenin. You can use acid, you can use alkalis, you can try some enzymes, but it does not cause any harm to it. Okay, then on this exine I have mentioned a small region called as germ pore. Pollen grain consists of a germ pore, and that part of the exine is called as germ pore where sporopollenin is absent. Okay, we said exine is made up of sporopollenin, but only in this region where sporopollenin is absent in exine, for that region we call it as germ pore. Okay, so in an exine itself, one portion is absence of sporopollenin. Because why it is absent? Because here pollen grain will start to germinate to form a pollen tube. To form a pollen tube, if it is completely made of a sporopollenin, completely filled with the sporopollenin, then pollen tube could not germinate. It could not come out of the pollen grain, because as we said, it is very hard, resistant and all. So that's why pollen tube could not come out, nor pollen grain can germinate. So that's why one portion of the exine will be not having sporopollen in. That portion we call it as germ pore, through which pollen grain is getting germinated. I have shown germ pore here. Not necessary it will be present here only. It may be present anywhere on the any side of the pollen grain, and there you have to show that there is a presence of germ pore. Okay. Then you have about intine. Intine is the inner layer. Inner layer, it is made up of cellulose, and this intine, it is having inside intine you are having two cells. One is generative cell, another one is vegetative cell. Vegetative cell is the largest cell. Generative cell is the smallest cell. Generative cell is floating in the vegetative cell. It is having smaller nucleus, it is having larger nucleus, and it is larger cell. Vegetative cell is also called as tube cell. Okay. Generative cell, it is used for the formation of gametes. Okay, generative cell, it is helpful for the formation of two gametes. Okay, we said pollen grain is a male gametophyte. If pollen grain is not a gamete, it carries a gamete. So like that you will find generative cell will form two gametes. Vegetative cell or a tube cell, this is used for the formation of pollen tube. Okay, pollen tube is formed from the vegetative cell. Male gametes are formed from the generative cell. Okay, so about this information you have in your textbook also. We shall see. This will be the explanation for your pollen grain structure. As you know, pollen grains, they are spherical, measuring about 25 to 50 micrometers in diameter. It has two layered wall. One is hard outer layer called as exine, which is made up of sporopollenin, and it is resistant to organic material, and it can withstand high temperature, strong acids and alkalis, and no enzyme that degrades sporopollenin is known so far. Okay, this is about exine. Then you have pollen grain. Exine has prominent aperture called as germ pore, where sporopollenin is absent. Pollen grains are well preserved as possible because of the presence of sporopollenin. As I said, pollen grains can be stored for a longer time in the form of fossils because of presence of sporopollenin. The inner wall of the pollen grain is called as intine. It is thin and continuous layer made up of cellulose and pectin. So intine is made up of cellulose and pectin. One mark question they have asked, or entrance exam question it is. In intine is made up of what? Cellulose and pectin. The cytoplasm of pollen grain is surrounded by plasma membrane. When the pollen grain is mature, it contains two cells. Which two cells? One is vegetative cell, another one is generative cell. Vegetative cell is bigger. It has abundant food reserve and large irregular shaped nucleus. Okay, it does not have particular shaped nucleus, irregular shaped nucleus. It is a large cell and it has large amount of food reserve. This large amount of food reserve is used by the generative cell to divide and to form two male gametes. Okay, the generative cell is small and it floats in the cytoplasm of vegetative cell. It is having spindle shaped with a dense cytoplasm nucleus, spindle shaped nucleus and with a dense cytoplasm. Okay, in over 60% of angiosperms, pollen grains are shed at two cell stage. What are these two cell stage? Okay, now pollen grains which are shed at two cell stage. Now you can find two cells. Which are those? Vegetative cell and generative cell. In 60% of angiosperms, pollen grains are fallen. Shedding is nothing but that falling. Okay, that after dehiscence, pollen grains will move through pollination. It might be because of insects, pollinating agents are there, might be because of wind, might be because of water. They move from one place to the another. Now that agents, when that pollen grain is getting shedded, it will be having two cells, vegetative cell and generative cell. It is called as two cell stage. Okay, one more, we have three cell stage. In three cell stage, what you have? In three cell stage, vegetative cell and generative cell. Generative cell, what I said, generative cell is used for the formation of two gametes. In some of those plants, before shedding, generative cell will divide and produce two male gametes. Okay, in some of those plants, generative cell will divide and form two male gametes when it is present in the plant itself. Before shedding, this takes place. The one who's before shedding, when it takes place, then it carries three cells. Okay, two male gametes and one vegetative cell. That we call as three cell stage. In few of the plants, what happens? Only two cell stage will shedded. When does that generative cell divide? That generative cell divide when that pollen grain has been reached to the stigma. Then this generative cell will divide to form two male gametes. Okay, where this occurs? This occurs in two cell stage when they are reaching it to the stigma. In three cell stage, they are present in the plant itself. Before shedding from the plant, there only this division takes place. So it is three cell stage. So that has been mentioned here. In over 60% of angiosperms, pollen grains are shed at this two-cell stage. In the remaining species, the generative cells divides mitotically to give rise to two male gametes before pollen grains are shed. That is three-cell stage. Okay, hope you understood this. Fine. Now this will be the explanation for structure of pollen grains. Here they have shown diagram how it will be present. Pollen grain, this is the microspore, this one is the microspore tetrad. Now one of the microspore tetrad they have shown here, which is rich in cytoplasm and a nucleus, and then it starts to divide and it undergoes continuously changes in that, then it appears like this. Okay, so the structure of pollen grain you should know to draw and explain about it. That complete explanation is here, you can get that information. Okay, now pollen grains, they causes allergy. Many species of pollen grains they causes allergy. You might have noticed that many of the people after having the flower, then after smelling the flower, they will start to sneeze, right? You might have noticed. So pollen grains of many species, it causes severe allergy and bronchial afflictions, that means bronchitis, and leading to chronic respiratory disorders like asthma, bronchitis, etc. Okay, so these are caused by pollen grains also. And it is mentioned that Parthenium or carrot grass, Parthenium, its another name is carrot grass, which came into India as a contaminant with imported wheat. Okay, while importing wheat from the other countries, with the wheat we have got Parthenium, and which has been spread all over India. Now this Parthenium will cause allergy. Okay, the most common plant that causes pollen allergy is Parthenium or carrot grass. One more question you will find, you will get this. Okay, after this you have pollen grains are rich in nutrients. Yes, pollen grains, as they causes allergy, even they are rich in nutrients. So in some of the countries they are used as food supplements. Supplements are those which we, in place of having your food, we are having in the form of tablets. As rather than having vitamins through fruits, we are having vitamin tablets. So directly from vitamin tablet, what body needs, requires, that we are having in the form of tablets, that we call as supplements. Okay, you have protein supplements, you have vitamin supplements, you have energy supplements, many are there. But pollen grains, which are rich in nutrients, they are also used as food supplements in the form of pollen tablets. In Western countries, large number of pollen products in the form are used in the form of tablets and syrups. Okay, where in Western countries, pollen consumption has been claimed to increase performance of athletes and racehorses. Because it is rich in nutrients, it provides energy. So to increase the performance of athletes, that is sportsmen, and racehorses, for horses, for those also it has been provided to to increase their stamina and get the performance do well. So that these pollens have been used for as food supplements also. So they have shown some information regarding that pollen products, you can see all those. Okay, next we have about pollen grain, the viability of pollen grain. Viability means capable of working, feasible. Okay, viable means capable of working. So viable, capable of working, till what time it will be working. Okay, as you can find a flower will bloom, its viability is what, it might be for 1 day, 2 days, 1 hour, 2 hours. After that it will it might get contaminated, it will get some other things, and it will get damaged, right? Like that. Similarly here you will find pollen grains, they have viability till what time they are working, that we have here. The period for which pollen grains are viable is highly variable, which means pollen grain viability, it varies from one plant to the another plant, and to some extent it depends on the prevailing temperature and humidity, which means it depends also on the temperature, ambient temperature in the environment, and even in humidity. Some examples they have given: in some cereals such as rice and wheat, pollen grains lose viability within 30 minutes. Okay, so as soon as they are released from the anther, then these pollen grains will lose their viability within 30 minutes, which means within 30 minutes it has to get pollinated. If it reaches to the stigma, then only it will get germinated. After 30 minutes, if it reaches to the pollen grain, then it will won't germinate. Okay, this takes place in cereals like rice and wheat. In some members of Rosaceae, Leguminaceae and Solanaceae, these are the families, 3 families which maintain their viability for months together, which means these pollen grains can live for months together. After release from the pollen anther, these pollen grains can stay longer viable for months together, after 2 months, after 3 months, after 6 months, if it is reaching to the female part, that is stigma, and getting germinated. Then also it is capable of germinating. But of this rice plants, it will get germinated within 30 minutes. Okay, so similar to that, you might have heard about storing semen or sperms, and like artificial insemination. This you have in future classes, there I shall discuss. Anyhow, here you have pollen grains. We can store pollen grains for a large number of species by using liquid nitrogen at minus 196 degrees Celsius. Okay, for this we call it as cryopreservation. Okay, cryopreservation is what? Storing of pollen grain or semen, storing of pollen grain or semen at minus 196 degrees Celsius in liquid nitrogen. We call it as cryopreservation. Okay, storing a pollen grain or semen at minus 196 degrees Celsius in liquid nitrogen, we call it as cryopreservation, which means pollen grains and semen or sperms or ovas, it can be stored by using liquid nitrogen in this temperature, so that it is helpful for storing for a longer time. In this, you can find such a stored pollen can be used as a pollen bank, similar to seed banks in crop breeding programs. We can keep this pollen grains for many years together, 100 years, as many years you want you can keep. It won't get damaged, and whenever there is a need of that particular pollen, you can collect it. As you have banks where you are going to store money, you have seed banks where you are going to store seeds. Similarly, you have this kind of banks where you are storing this pollen grain, semen, ova, and many of the belongings. Okay, for that we call it as cryopreservation. Okay, this is about today's class. In today's class we have started with the microsporogenesis, right? We have discussed about structure of pollen grain, and then pollen grain, what does it causes, and uses of pollen grain, viability of pollen grain, and how it can be stored or preserved. Okay, so if you have any doubts in today's class, you can please comment it in the comment box, and I shall try to solve your problem. Okay, we shall meet in our next video. Stay safe, stay healthy.